Good morning, everyone. Today we start with about our new topic in Unit 1, ICH guideline. So in your syllabus, they include about the overview about the ICH guideline, which is quality guideline, safety guideline, efficacy guideline, and multidisciplinary guideline. And also in your syllabus, we see about what is meant by the stability testing and what they are including about the guideline related to stability testing. So this point we can see related to ICH guideline. So what is meant by the ICH? It is International Conference on Harmonization of Technical Requirement for Registration of Pharmaceutical for Human Use. So this is ICH. It is the name itself indicate International Conference on Harmonization to maintain the harmony throughout the world, throughout the internationally. So that's why the term itself, the ICH, due to it is the pharmaceutical requirement we directly use for the human or human purpose. So that's why it says as the International Conference on Harmonization of Technical Requirement for Registration of Pharmaceutical for Human Use. So this is meant by ICH, technical requirement, whatever we want for registration of pharmaceutical, which we directly use for the human purpose. This all these things, their quality, their safety, their efficacy, it's all these things which is come in the ICH. So we can see or we can go for this their overview regarding the quality guideline, safety guideline, efficacy guideline, and multidisciplinary guideline. So here you can see the brief overview of QSEM with special emphasis on Q series guideline. So for only the quality having the different series of guideline, safety guideline, efficacy guideline, multidisciplinary guideline, we can see one by one. So this ICH divided into the four category of their guideline: quality guideline, safety guideline, efficacy, and multidisciplinary guideline. So the quality which related to the stability, impurity testing, and good manufacturing practices. So we already see about what is meant by good manufacturing practices, from personal to the premises, equipment, our good manufacturing practice related to the packaging, our particular area or premises. It's everything which is comes under the GMP. So related to the each and every terminology which directly affect our quality. So the stability of our pharmaceutical product, whatever the drug is having some impurity or not, so impurity testing, profiling, it is also given in the quality guideline. So there are different specifications is which is given into the quality guideline. And our stability, we are having the overview about the stability guideline. So it come in a quality guideline. So safety guideline, either our pharmaceutical product, it is carcinogenic, they are causing some carcinogenicity to the human, some genotoxicity, some reprotoxicity, it is there or not. So the safety level to use for the human, it is there or not. These things we can take in the safety guideline. In efficacy guideline, how to conduct the clinical trial, their efficacy, how to take that particular drug product, their efficacy, it is there in this efficacy guideline. And pharmacogenomics, this is also come in this efficacy guideline. So this is the different guideline. You can see here we are having the quality related guideline, we are having the safety profiling guideline, efficacy of our new product, their clinical trial guideline, it is also there. Their pharmacogenomic profiling guideline, it is also there. And multidisciplinary guideline, we are having the MedDRA, CTD, our common technical documentation, electronic standard. So for that, how to filing that CTD, eCTD, what is in my MedDR. So everything, the instead of quality, safety, efficacy, whatever the thing which is remaining, which comes into the multidisciplinary guideline. So this is the ICH guideline, which is divided into the four disciplines, which is quality, safety, efficacy, and multidisciplinary guideline. So we can see the different Q guideline, like quality Q1, which is for the stability guideline, ICH Q2A, which is analytical validation guideline, ICH Q3I, which is the impurity guideline, Q4, which is pharmacopoeial guideline. Q5 for the quality of biotechnological product. Q6, which is in the specifications. Q7, which is good manufacturing guide for active pharmaceutical ingredient. Q8, which is pharmaceutical department, how to conduct all the activity now pharmaceutical department related to. We are having the Q8 guideline. For Q9, we are having quality risk management guideline. In our QBD, we can see different specification, different quality related guideline into it. Also, Q11, we are having the development, manufacture of the drug substance, chemical entity, biological and biological entity. Q12, which is life cycle management guideline. So we are having, you can see here, the Q1 guideline for the analytical validation, we are having in unit 5 separate analytical validation. For that, we can refer ICH Q2 guideline, ICH Q3 for the impurity guideline, Q4 for the pharmacopoeial guideline, Q5 for the biotechnological product, Q6 specifications, GMP guideline for the API. Whatever the pharmaceutical company who is manufacturing the API, they can refer the ICH guideline because this is the international standard. So that's why we can refer this Q7 guideline for the guiding us for manufacturing of our API. Q8 for the pharmaceutical development, Q9 for the quality risk management, pharmaceutical quality system, Q10 and Q11, which is the manufacturing of the substance, chemical entity, biological and biological entity. Q12, which is life cycle management. So for QBD, we are having quality by design approach. We can refer this the specification Q6, quality risk management Q9, quality system Q10 guideline and Q12 life cycle management guideline. Okay, so this is our quality guideline. Also, in the quality guideline, we are having the different uh, Q Q1A to QF, which is for the stability guideline. So in Q1A, we are having the stability testing for the new drug substance and product. Q1B, which is for the stability testing of photo stability testing of new drug substance and product. Q1C, which is stability testing for new dosage form. Q1D, which is bracketing and matrixing design for stability testing of new drug substance and product. Q1E, which is evaluation of stability data. Q1F, which is stability data package for registration application in climatic zone 3 and 4. So this is the stability guideline for the Q1 to QF, having the different classes like for new drug substance, testing of new drug substance, whatever your project, if you are performing any project for formulation of new product, so that for the new drug substance you can follow or you can refer the Q1A guideline. For photostability testing, the drug which is having some photosensitivity, for that purpose the new drug photostability testing, we can go for the Q1B guideline, ICH Q1B guideline for that particular API and particular product. Also for the new dosage form, like we are formulating that particular new dosage form, after that we can check their stability. For that purpose we can refer Q16 guideline for that particular dosage form, the newer dosage form. The bracketing and matrixing design for stability testing of new substance and product, like we can perform the bracketing method or matrixing design for the stability, checking the stability of that particular product. So we can refer the Q1D guideline. Q1E, which is evaluation of stability data. How to evaluate whatever the thing we are performing, there is need to be evaluation. So that evaluation of that stability data, whatever the 3 month, 12 month, 4 year study, we are having the different period of the stability study of new product or new drug substance, for so that whatever the result we got accordingly, we need to evaluate our final evaluation. For the evaluation, how to evaluate that study, we can refer the Q1E of the stability data. Q1F stability data package for registration application in climatic zone 3 and 4. So we are having the different, we can see, because in your syllabus they include about all about the stability overview regarding the stability study, ICH stability study. So in that we can refer about what the in descriptive way, what is the Q1A for this, what is the photostability study, how to perform that for the bracketing matrixing method, evaluation of stability data, and their different climatic zone, how we classify the different climatic zone. So zone 3, what is their humidity condition, what is their temperature conditions. So these things we can having in this stability data pack. So we are having Q1A to Q1F stability guideline in this quality guideline. Also, we are having the Q2 ICH Q2 R1 quality guideline for analytical procedure, text and methodology. So we are having the different analytical procedure. In that, we are having the text for how to go for this accuracy, precision, how to calculate this linearity, accuracy, precision, all these validation parameters, how to develop the analytical method, till how to get this validation of analytical procedure according to the different text is there, different methodology and their procedure and their specification, everything which is included in ICH Q2 R1 guideline. So ICH Q2 R1 guideline in our analytical validation topic, we descriptive way we can refer this how to go for this analytical validation. Okay, so in that time we can refer this Q2 R1 guideline. Also, we are having the guideline for impurity. So whatever the new drug or new drug product, we are having the Q3A to Q3D impurity guideline. So Q3A, which is guideline for this impurity in new drug substance. Q3B, which is impurity in new drug product. Q3C, impurity guideline for residual solvent, and Q3D, impurity of guideline for elemental impurity. So in you can see here the drug substance, drug product, you know how what is the synthesis method, you can refer for this particular chemistry you can use, or whatever method you can use for synthesis of drug substance. If there is chances, whatever solvent you are using, there is chances for the impurity, so you can go for the profiling that impurity in new drug substance. Also, we are having the drug product, so we can go for profiling that impurity in drug product. Q3C, which is guideline for residual solvent. Some residual traces it is there in our product. So we can find it out by referring this guideline, how to find it out, how to make the procedure for this residual solvent. Q3D, which is for the elemental impurity. So this is the different Q3A to Q3D impurity guideline. Also in quality series, we are having Q4 guideline for pharmacopoeial specification, pharmacopoeial harmonization. In our ICH, there is having the different area, like we are having the Indian pharmacopoeia, European pharmacopoeia, European pharmacopoeia, Japanese pharmacopoeia. So we are having the different pharmacopoeia. For worldwide, we require the same drug product, their same synthesis, their same monograph in different pharmacopoeia is maintained, their quality is maintained, their harmonization throughout this world. So that's why they are having specification for designing that particular pharmacopoeia. So for that we can refer for this pharmacopoeial harmonization, we can refer the quality 4 guideline A and quality B guideline 4B guideline for the evaluation and recommendation of pharmacopoeial text for use in the ICH region. So whatever the region we are having, the text for the evaluation and recommendation of pharmacopoeial text, which is there in the pharmacopoeia guideline. Also Q4B annexure. So we are having some annexure for 2R1, which is extractable volume of parenteral preparation general chapter. So for this pharmacopoeia, we are having some annexure. If you see the pharmacopoeia, you understand there is having some annexure. So Q4B guideline, how to write that general chapters for the parenteral preparation, which is given in the Q4B guideline. Q4B annex treat R1 guideline for test for the particular contamination, subvisible particle general chapter. So this is the contamination and visible particle general chapter, which is given in the Q4B guideline. So this is our Q3, Q4 guideline. Also Q4B guideline for annexure 4A-R1 for microbiological examination of non-sterile products. So parental product, we are having the annexure 4A-R1. In that, we are having the different guideline for the annexure 2 R1 guideline and annexure 4 2 R1 guideline, and also we are having the 4A-R1 guideline for this non-sterile product microbiological enumeration test and general chapter. Q4B annexure 4B-R1 for microbiological examination of non-sterile product test for specific microorganism general chapter, which is there in Q4B guideline. Q4C guideline R1, you can see here the A, B, C guideline for this. So we already see Q4A guideline, Q4B guideline, Q4C guideline. So Q4B guideline for this non-sterile product test and specification of this given in the general chapter. The microbiological examination of non-sterile product, acceptance criteria for pharmaceutical preparation and substance pharmaceutical use. So preparation, how to prepare the substance, their accept according to their acceptance criteria, which is given in the annexure Q4B 4C R1 guideline. So this is the annexure. If you see the pharmacopoeia, then you understand the different annexure in their different chapter it is there. So accordingly, for sterile product, we are having the different annexure. For this non-sterile product, we are having the different annexure. For preparation of the solution, preparation of the preparation and substance for pharmaceutical use, it is having the different annexure. So this is the different annexure we are having in pharmacopoeia. So for that writing specifications, right? So we can refer this quality guideline. Q4B Annex 5R1, it is disintegration test general chapter. So this tablet, it is important part in our pharmaceutical. So this tablet dosage form, so we can refer this disintegration test chapter. Uniformity of the dosage unit, which is given in the 6th chapter R1, Q4B 7R2 for the dissolution test general chapter. So what is the dissolution apparatus? What is type 1, type 2, type 3, type 4, type 5, type 6 dissolution test apparatus? How we go for this different dissolution media, how to prepare that dissolution media, how to perform their calculations, how to maintain the sink volume. So what is their specifications to that particular dissolution test apparatus? Each and every description which is given in the dissolution test chapter. And the dosage unit dosage form, you can refer the uniformity calculation in this 6R1. 7R2 we can refer for the dissolution test chapter. 8R1 stability test, how to perform the stability test for particular product, which is given in the pharmacopoeia also. You can refer the pharmacopoeia as well as ICH guideline. So in the both way, you can go for the stability test chapter, how to perform the stability test according to ICH. It is also given in the pharmacopoeia. So whatever the drug, it is IP specification, USP specifications, or BP specifications, you can refer this stability test chapter. Q4B Annex 9R1, which is tablet friability general chapter, and also Annexure 10R1, Q4B 10R1, which is polyacrylamide gel electrophoresis. So electrophoresis method by using polyacrylamide gel, how to perform that electrophoresis, which is given in the pharmacopoeia, the general chapter, which is 10R1. When you refer the Annex 11, which is capillary electrophoresis general chapter. So we are having the polyacrylamide gel chapter, capillary electrophoresis chapter, analytical sieving general chapter in Annex 12. The Annex 13, we are having bulk density, tap density of powder general chapter. So how to go for the bulk density, tap density, which is also given in our pharmacopoeia. BT test, you know the parental formulation, how we perform for the BT test, the bacterial endotoxin test, which is also given in this, which media you can use, how to go for this bacterial endotoxin test, their specifications, everything which is included in Annex 14. So this is our quality 4 guideline. After that, we are having the quality 5th guideline, quality 5th Q5AR1 for the viral safety evaluation of biotechnological product derived from cell lines of human or animal origin. So Q5A, Q5B, which is quality of biotechnological product, Q5C, which is quality of biotechnological product and quality of biotechnology substances. Biotechnology and biotechnological product, their quality we have to maintain. So this specification you can refer in the guideline Q5C guideline. For the Q5D guideline, we are having the derivation and characterization of cell substrate used for the production of biotechnological and biological products. So we are having for this quality maintaining in biotechnological product, because we can use the whatever the biotechnological product, it is directly derived from the cell line or animal origin structure it is there. So that's why their quality maintenance, it is not so easy task. For that, we are having the separate guideline regarding each and every point. Like you can see here, the safety evaluation of biotechnological product derived from cell line of human or animal origin. Q5B, quality of biotechnological product. Quality of biotechnological product, it is also given in the Q5C guideline. Also, the derivation and characterization of cell substrate used for the production of biotechnological product. It is their derivatization, why we use only this strain. It is also given in this Q5D guideline. Q5E guideline, which is comparability of the biotechnological products subject to change in their manufacturing process. So whatever the comparability of biotechnological subject to change their manufacturing process, if we require to change our any manufacturing process, so we can refer this Q5E guideline for the biotechnological product. Also, we are having the quality 6 guideline. There is having some specification. The specification regarding the test procedure, acceptance criteria for new drug substance and new drug product. So their chemical substance, we are talking about the specifications related to the chemical substance, test procedure and acceptance criteria for new drug substance and new drug product. So this is given in the Q6A guideline. For Q6B guideline, specifications related to the test procedure and acceptance criteria for biotechnological product. So for this new drug substance and biotechnological product, we are having different specifications which is given in the Q6A and Q6B guideline. In Q7 guideline, we are having the GMP guideline for active pharmaceutical ingredient, Q8R2 guideline for pharmaceutical development, Q9 guideline for quality risk management, Q10 guideline for pharmaceutical quality system. So this 3 guideline in different way we can see when we want to apply the quality by design approach in our pharmaceutical product. So you can refer how to develop that product, what is the risk management factor in that particular product, and what is the pharmaceutical quality system. So this everything which is we can refer for going to apply any quality tool in our pharmaceutical product. So this guideline, Q8 guideline, development guideline, pharmaceutical quality risk management guideline, Q9 guideline we can refer, Q10 guideline related to the pharmaceutical quality system, Q11 guideline for development and manufacture of the drug substance like chemical entity, biotechnological entity. It is given in the development and manufacturing of our pharmaceutical substance. It is given in the Q11 guideline, and Q12 guideline which is there in the life cycle management guideline. So this is the different guideline till we can see Q12 guideline. So this is all about our safety, our quality guideline. After that, we are having QS guideline, S for the safety guideline. So safety, for that we are having, you can see here S11 guideline. Till S11 we are having the different guidelines. So for safety guideline, S1A for carcinogenicity, S1C study for this study guideline, S2 for this genotoxicity study. So we see already the carcinogenicity, genotoxicity study S2 guideline. We are having the toxicokinetic guideline S3A to S3B, which is toxicokinetic guideline. S4 for pharmacokinetic toxicity. S5 for the testing for this pharmacokinetic toxicity or safety testing. We can say it is given in the safety 5 guideline. Safety 6 guideline for reproductive toxicity. So the toxicity which is reproductive in different way. So that's why we can refer if this type of the problem it is arise in our pharmaceutical product. So we can refer the S6 guideline, S7A to S7B. It is biotechnological product related safety guideline which is given in the S7A, S7B guideline. S8 guideline for the pharmacological study. So different pharmacological parameter, their safety we can refer the safety guideline S8. For S9 immunotoxicological study in pharmaceutical non-clinical evaluation also given in this safety guideline S9. S10 which is for the uh, non-clinical guideline, and S11 which is the safety guideline. For pediatric patient non-clinical guideline which is given in the S10 and S11 which is the safety guideline. So this is after that the quality 12 guideline, and their subpart we see after that we are having the safety guideline like carcinogenicity, genotoxicity, toxicokinetic, pharmacokinetic, reproducive toxicity, biotechnological product safety related to pharmacological study, immunotoxicological study, non-clinical evaluation of this anti-cancer photo safety evaluation, and non-clinical pediatric evaluation study and safety study. So this is overall the safety guideline which is given by the ICH. So we are having only the brief overview. So we are not going in detail regarding how to calculate the carcinogenicity, genotoxicity, pharmacokinetic, pharmacological study for the safety level. So this is we are not going to in detail. We just having the brief overview. So you must know about what is the guideline which is covered in this different area. For efficacy guideline, we are having the clinical safety for the drug used in long term. So for the efficacy guideline, we can check whatever the product we are formulating, they are efficacious or not. So we can go for the clinical safety for the drug used in long term. So we can check their clinical safety by following or referring E1 guideline. For efficacy guideline, it is clinical safety for drug used in long term. E2A for treatment and pharmacovigilance guideline. Those who go in the pharmacovigilance, they refer the E2A guideline and E2F guideline for the clinical study. How to maintain the report and doses, which is they can refer the E3 guideline, efficacy guideline. E4 for response study, the ethical factor code. So whatever the ethical factor we are using in clinical study, how much the subject we want to enroll, what is their ethical practices, their risk to that particular clinical study. So each and everything, ethical practice code, ethical factor, how to decide. So each and everything which is given in the efficacy E5 guideline, E6 guideline for the good clinical practice guideline. So how to go for the clinical trial, what is the good clinical practice guideline. So those who want to go for the clinical trial study or clinical safety study, or they want their career in the clinical trials or clinical research. So in that you must know about what is the E6 guideline they can given. So E6 for the good clinical practice guideline. In that we are having the different clinical study and clinical safety for the long-term treatment. How we decide this given in the E1 guideline. E7 guideline, the clinical trial in geriatric population. E8 guideline for the general consideration for clinical trial. Their trial statistical principle for clinical study. Their statistical, whatever trial, whatever result we know with their statistics. So we can go for this E9 guideline. Trial choice of control group, which is given in the E10 guideline. E11 for the clinical trial and clinical trial in the different population, which is given in the E11 guideline. E12 for the pediatric population in the clinical guideline, which is given in the E12 guideline, E14 for evaluation by therapeutic category, E15 for clinical evaluation of Q10, E16 for definition of pharmacogenetics and qualification of genomic structure, their efficacy of genomic area or genomic formulation. We can go for that particular product for their efficacy testing by referring the E17 guideline. E18 for biomarker multi-regional clinical trial, E19 for trial in genomic sampling, safety data collection, which is given in the genomic sampling data trial, their safety data trial, which is given in this E19 guideline. So E17 for qualification of the genomic label, their biomarker, their multi-regional clinical trial, which is given in the E18 guideline. So you can see here Q series having the 12 guideline and different sub guideline. Our safety guideline, it is also 11 guideline, and our efficacy guideline, you must know, remember about the till the 19, whatever the part they are covered in this efficacy guideline E19. Also we are having the multidisciplinary guideline. For the M1 guideline, we are having the medical dictionary for this. So this terminology which is given in that particular medical dictionary, which is given in the M1 guideline. M2 guideline for the electronic standard, whatever data we are covering in our pharmaceutical industry, that electronic standard for that new drug application, ANDA application. So whatever the regulatory requirement, like whatever the electronic standard we want to generate or submit to different regulatory agency, which is given in this their standard, which is given in the M2 guideline. M3 guideline, the non-clinical safety study. So whatever the safety study in the non-clinical which is there, this non-clinical study which is given in the M3 guideline. M4 guideline, the common technical data. So whenever we want to file our CTD by the electronic way or eCTD, there are different modules. So there are different modules is having the different area. So that point how to submit all this technical document into this their guideline. We can refer the M4 guideline. For M5 guideline, we are having the element and standard for drug dictionary. So whatever the drug dictionary we are having, so whatever the standards we are having, M1 is only the terminology which is given. So whatever the dictionary is having the terminology, it is given in this, and also the data and standard of that particular dictionary which is maintained by the M5 guideline. M6 guideline, which is gene therapy, how to go for the gene therapy, what is their purpose, what is their advantage, disadvantage. So everything which is given by the M6 guideline. M7 guideline, they include the genotoxicity. M8 guideline, the common technical document. So electronically we can submit the common technical document. So accordingly, whatever the common document and common technical document electronically we have to submit. So there are different modules, there are different specifications which is given in this M8 guideline. M9 guideline for biopharmaceutical classification system based biowaiver. So how this biopharmaceutical classification, class 4 classification we are having according to their solubility and their particular lipophilicity. So we are having the drug, it is having low soluble, high permeable. So accordingly, we are having this classification of biopharmaceutical classifications. So this classification which is given by referring this M9 guideline, M10 guideline, the bioanalytical method guide validation. So we are having the analytical method validation. In that we can go for this according to the solubility of that particular drug substance. We can go for the solvent selection accordingly. But in bioanalytical method of validation, we can select their solvent, like our biological solvent we can use for this method validation, method development. So all this bioanalytical method validation which is given in this M10 guideline, and our analytical method validation which is given in the quality guideline Q2 R1 guideline. So this is the different 4 guideline given in the ICH. In this lecture we study about what is meant by quality guideline, safety guideline, efficacy guideline, and multidisciplinary guideline. So we can stop here. In our next session we can start with what is meant by the stability study. So I hope you all are understand about what is meant by the ICH, and we already discussed about what is the quality guideline, safety guideline, efficacy guideline, and multidisciplinary guideline. Okay, so we can stop here. Thank you.